Good afternoon. I'm Baekhyung Song, working at KSBM in Germany and University of Science and Technology in Korea. I would first thank the conference organizers for inviting me for a lecture. It is my great honor to deliver a talk in the meeting of Korean Society for Biomaterials. Today, I'd like to briefly introduce some examples in soft matter technology, which cover mainly polymer microgels and biomicrofluidics, and are potentially applied to the future development of biomedical microdevices. Let us begin with an overview on the drug delivery through the skin. If we consider the skin as the drug delivery route, sometimes it becomes critical to choose the best location for the drug administration. And the skin impedance can be a good parameter for such non-invasive sensing because low impedance loci may indicate the most hydrated local points that facilitate the drug diffusion. After determining where to inject, it is needed to select the type of injection method. For example, we may consider a hollow microneedle array device for controlled long-term infusion as a patch format. And then choosing an appropriate drug carrier is also a critical factor. We have many various types of drug nanoparticles or drug-loaded microgels. For the last, after the administration, the transport of drug carriers needs to be monitored using in vivo fluorescence imaging, for instance. So in this way, an entire cycle of diagnosis, therapeutics, and physiological monitoring is completed. Now, I'd like to quickly introduce my works corresponding to each step of the whole process. For the miniaturized drug carriers, let us start by introducing microscale hydrogels, referred to as microgels. A microgel is a microscale 3D network of polymer chains with mostly spherical morphology, which can be formed by dissolving polymer powder in a solvent with cross-linking, and it can be in a swollen or dried state. Its size can be controlled in diverse ways as compared to that of a sesame grain here as a scale reference. Since more than 90% of the microgel volume is solvent water, smaller particles, macromolecules, or even living cells can be encapsulated in a microgel. With this regard, a microgel can be used as a miniaturized scaffold for the cell function control to operate as a microscale extracellular matrix for tissue mimicry. Since the ECM-derived polymers like collagen and gelatin contain cell-binding motifs, the embedded cells can interact with the polymer network to feel mechanical feedback from the gel matrix. This process can regulate the cell metabolism to secrete therapeutic molecules or proliferate, differentiate into a desired cell type as a response to external stimuli. Moreover, when the gel matrix has an ordered structure, the anisotropic substrate of liquid crystalline scaffolds provides tunable topography and stiffness, which are capable of inducing directed cell migration and growth through biophysical interaction between the cell and substrate to enable versatile applications for tissue engineering. So there is a high opportunity to use microgels as 3D mimics, enabling cell microencapsulation with fine-tuned cell matrix interactions. The embedded cell fate regulation can be achieved by tuning the artificial ECM component, the gel elasticity with varying cross-linking degree, as shown in this figure, and matrix degradation by enzymes from the laden cells. For the biofabrication technique, the droplet microfluidics has been widely utilized for the generation of cell-laden microgels, as depicted in this figure, where we have manufactured the microfluidic chip using 3D printing methods. After fabricating the cell-laden microgels, mesenchymal stem cells in this case exhibit increased interaction with the gel matrix with time. Let us have a more close look at the on-chip 3D microgels for the application to hepatic tissue mimicry. Through an integrated channels and reactors design, the derivative formation, gelation, and collection processes are automatically controlled to generate monodispersed gelatin droplets with even number of embedded cells. Then after cross-linking, microgels can be microfluidically manipulated on chip. Also, we see that this miniaturized liver mimic can be an artificial niche of cells for supporting long-term cell culture. On the other hand, the microgels can be used for controlled drug release, which can be either thermoresponsive and magnetic field responsive designs. In this example, a gelatin microgel was fabricated to trap small amount of thermosensitive pinipan chains in the cross-linked gelatin matrix. When the temperature increases above the critical point, the entrapped pinipan chains collapse and the whole gelatin matrix collapses, which is not thermosensitive by itself, so that the embedded drug molecules are squeezed out by microgel diswelling. This type of microgels can be further functionalized with supermagnetic iron oxide nanoparticles to enable remote controlled drug release upon alternating magnetic fields. Furthermore, similar magnetic field responsivity can be conferred to the nanogels that are developed to penetrate inside target cell body. In addition to these simple magnetic nanoparticles like in this image, more complex nanostructures can be developed to support new therapeutics. For example, the polycation-induced coil globule phase transition of a DNA chain is one of the major principles for packaging long DNA chain in a nanoscale space. In principle, a single DNA chain is a strongly charged and semi-flexible polymer that exhibits a coil conformation in solution. However, upon addition of polycations, the chain drastically collapses into a nano-sized globule. Based on this phenomenon, a DNA toroid is formed inside a virus capsid, which is in a liquid crystalline phase. This is a powerful way to compact large amount of genetic information in a tiny space and is utilizable to gene delivery strategy such as cell targeting adenovirus vector. Once a drug or gene carrier type has been chosen, it is to determine where and how to inject such carriers in vivo. For this purpose, a microneedle-based electromechanical device has been developed for transdermal drug delivery. The core idea in this device context is to use a metallic hollow microneedle as an electrode and needle at the same time, where circuits for electric impedance sensing and electropulsing are coupled independently. So first, we perform skin impedance probing in a contact mode, then subcutaneously inject microliter volume of drug carriers at an appropriate location by connected pump, and apply transcutaneous electric stimulation for facilitated drug diffusion. Furthermore, subsequent monitoring of the diffusional behavior of the model drug carriers can be followed in vivo. Throughout the slow infusion process, fluorescence real-time imaging showed that the dispersion is more and more diffusing in the skin with time, so that we could quantitatively characterize the diffusion kinetics with mathematical models. Then, what will happen to the injected nanodrug carriers in the body? Using a similar but more sophisticated imaging setup, we can monitor how they are transported through the lymphatic system. Because the lymph is a complex fluid, the colloidal stability of the injected nanoparticles usually decreases, so that we see micron-sized aggregates coexist in the nanofluid inside the lymph vessel. Utilizing the suspending aggregates as tracers with a high fluorescence contrast, we can visualize the microfluidic behavior of the nanofluid during the peristaltic cycles of a single lymph vessel. Given that the drug carriers are injected and followed in vivo, then we need to check the physiological change induced by the drugs. For instance, monitoring the body fluid viscosity of diabetes patients may be critical for fine diagnosis. For this purpose, here we use a transparent and hollow microneedle device that enables the biofluid viscosity measurement with a simple and rapid in situ optomechanical technique. The microneedle is injected to a soft tissue and a slightly negative pressure is applied through the hollow needle to obtain a nanoliter volume of biofluid, where we can trace the Brownian motion of suspended microcells and debris. And the viscosity value is directly deduced from the 2D Stokes-Einstein equation. Thus, the in situ optical viscosity monitoring becomes to be easily automized with a minimal volume of the biofluid. Finally, the tissue and cell level effects of the administered drugs are estimated with soft tissue characterization such as TM, SEM, tissue section, and histological staining according to the typical protocols in histopathology. In conclusion, we had a brief overview on a research cycle of biomedical microdevices, which is comprised of microgels and nanoparticles, microfluidic chips and biodevices, in vivo monitoring with bioimaging, diagnosis in tissue and cell levels, and then back to the improved design of the microgels and nanoparticles. I would emphasize here that the soft matter science and technology play a central role in this helical cycle of device development. In this last page, I would acknowledge our group members and collaborators, especially Professor Andreas Mans and Professor Leon Abelman, and our partners and funding sources. We are currently open for students and postdoc positions, and thank you very much for your attention. And it would be my pleasure to have your comments or questions.